map of the different places where the microbiometer, look, there's Anchorage, Alaska right there, up there. That's me, I guess, huh? And about 10,000 other people. No, and here's across the United States. Here's down in South America. Here we are. Hey, you even got some down there in Africa. This is really terrific. Australia, quite a few people in Australia, New Zealand, Tasmania. Unbelievable. So Judith and I were talking about this map. I'm going to see if I can't, and I'll switch it back here. Judith and I were talking about that map. And, you know, we were talking about the fact that when I get a reading in Anchorage, Alaska of, let's say, 400 or 500, you know, that's a great reading and I'm excited. But if you live in Biloxi, Mississippi, and you get a reading of 400, you should be jumping up and down and doing handsprings because it means something, a slightly different. And so Judith and I wanted to talk a little bit about what does microbiomass really mean, and how do you use microbiomass as a tool in order to be able to make you a better grower, gardener, whatever you happen to be. All right, so let's go back to that map. We're going to talk about the biomass in a minute, but the fact that you've got all of these different locations on that map of where people have purchased and are using the microbiometer, is that a growing list? Are you seeing it spread like crazy, or is it pretty much static these days? It's
yeah, definitely. A lot of users in Australia and New Zealand, and we've got new users in South Africa, Israel, Kuwait, Brazil, and Chile. If it weren't for the pandemic, we'd have a lot more users there right now. There's trouble getting things in and out.
Sure,
you know,
absolutely.
It's like,
well, now
when
those readings are made, it seems to me that at some point in time, we should be able to, as a group of users, be able to use the readings that we give you voluntarily to really perfect different ways of using the microbiometer. I mean, is that something you guys have in the back of your mind?
No, that's the goal. As we get more users with more different soil types, we can use the location and their readings and some of their, if we know what their practices are, then we can start to actually, I don't want to say make recommendations, but at least give better ranges of where they should be.
Absolutely. So,
you know what I mean?
Like capability technically, because you're the tech guy, at some point in time to be able to overlay different things. I think they call it GIS. Overlay different things into your technology, so that, for example, I'm in Anchorage, Alaska. I get a reading, but I can also look at what my soil type should be here, and therefore what my reading. So that's what's potentially could happen.
Right. That's what's coming along. We're working very hard for that right now.
Why that's important? It's because there are 5 or 6 other different tests that will tell you what the microbiomass is in your soil. Whether they're more expensive or cheaper, that doesn't make any difference. The point is, there's so many different kinds, and because these are much more expensive than what you guys do, it's hard to get a standardized reading. So, for example, the reading of biomass that I get using the XYZ test system versus the one you might get using the ABC system, we both get a number, but it's hard to correlate that number. Whereas with the microbiometer, you're going to be able to say, I live in Biloxi, my 200 is comparable to, whatever. I mean, it's the same number. Yeah.
A 600 numbers. Right.
Yeah.
Right. It just depends.
Yeah. Well, Jeff,
turn you over to Judy now. Hold on.
I'm just saying, like, one of the reasons we started on this is you know, having been in medical diagnostics, when you send your sample into the lab, they get a certain range on their tests. Now they just adjust their reading to read what, you know, to make it look like it's supposed to be according to the literature. But that doesn't mean that's exactly what they're reading, because different tests will give you slightly different readings. So we want to be able to give people the range for their area. That does not mean that you can't improve it. Okay, so you can improve the range by the type of fertilizer, or sometimes people even bring soil in. I was at one farm where they just brought in a whole 6 inches of the whole top acre of soil.
Right.
So obviously that changes things tremendously. But there is, you know, generally speaking, based on the type of soil you have a limitation on what you have. And I think your readings were more high, like 600 to 800.
So I noticed that my good friend Tad from this organization joined him. You know, the soils that he's got versus my soils versus your soils in New York, you know, there is a range. And I think we should really discuss that range, because I think people who are in, again, I'll use Biloxi, Mississippi, if they get a 300, that's a great number for them. But does that translate into their being able to grow great cannabis, for example, comparable to my 600 or 800 number? Do I grow this? Is it the same sort of nutrient capability in the soil, even though we've got two different numbers? And we were talking a little bit about that, Judith, and it comes down to, I think, the carbon nitrogen ratio and the microbiology.
Right. Well, the soil, you know, what we do is we correlate very well with the soil fertility. Okay, that big study at University of Tennessee, we were the only ones who actually were able to correlate with soil fertility. The Cornell Soil Health Panel didn't correlate with their soil health. Neither did the Alabama or the Haney test or PLFA or any of them. And we correlate very well with soil organic carbon.
Wait, you got to stop there, because
and there's a reason
for that. Put this in every soil organic carbon. There's different kinds of carbon in the soil, right? When you say soil organic carbon, is this available carbon that microbes can use?
That's the one of the big issues that you see when you read the literature. So to roughly break it down like Kirkby did, there's stored organic soil carbon, and that's what is limited by your soil type, because the organic molecules will attach to different metals and to different clays. So depending on the structure of your soil, there's a certain amount of soil carbon that you can store. And then there's like more fresh organic matter, which hasn't been broken down quite so much, and it is not, you know, really tightly associated to minerals and clay, and that's much more available to the microbes. So how does this go? Well, you talk all the time about exudates. So the plant, when it wakes up, puts out exudates, and the microbes, you know, usually we see almost a doubling of the microbes at that time. But if there's not more food there, because the plant has just given carbon, sugar, and some amino acids, if there's not enough nitrogen, phosphorus, potassium, sulfur, etc., those microbes can't support that growth and they fall back. Okay, so that's what we mean by bringing up the fertility of your soil for your crop. Means that the fresh organic matter, or the more available organic matter, has to be there ready for those microbes to use, if the microbes are to be the way that you're feeding your plant. If you're using chemical fertilizers, you're bypassing the microbes. You know, it's just like giving somebody their food IV.
Because what you're really talking about is a carbon nitrogen ratio, and this is really sort of key. And I think a lot of growers and a lot of gardeners don't get this. It turns out that in the soil, you and I both know that there's a sort of an average, you know, some people go, okay, the bacteria have this carbon to nitrogen ratio, and the nematodes have this carbon to nitrogen ratio. But in fact, if you just take the United States government statistics, it's about on average 8 carbon to 1 nitrogen for microbes in the soil. And it turns out that the best soil for growing crops, now this is not necessarily trees, this is not your shrubs, this is not perennials, but for growing crops, that would be lettuce, potatoes, cannabis, hemp. What you want to have is a carbon to nitrogen ratio of 24 carbons to 1 nitrogen. And that number breaks down to the microbe in general needs 16 carbons in order to live. In other words, to its energy, and 8 carbons, which makes the up to 24 for maintenance, for repairing walls and, for fixing things that get broken, etc. So 24 to 1 is what the soil needs to be. And so when you put in fresh material, the microbes react in terms of that 24 to 1 number. If the material you put in, and I'm going to stop in a minute, Judith, so you can jump in here. If the materials you put in to feed those microbes has a ratio below 24 to 1, then it gets broken down and eaten up quickly, because in order to get the carbon, they got to, and if it's above 24 to 1, it's a whole different thing. And so when you, and in fact, somewhere around here, let me see if I even have it. I mixed it up.
I think you kind of mix it up because if it's too rich in carbon and there's not enough nitrogen or anything,
right?
That's sort of like feeding sugar to your children.
Give an example of that. That would be.
So what happens is
like rye or corn stover or oat straw. 71. Oat straw is 70 to 1, as opposed to manure, which is like maybe 17 to 1. So what should you put on your garden? Manure or oat straw? Well, people put manure down. Well,
you want to mix them up, for a large part. And then of course, people, especially like for corn and things that are big, nitrogen hogs, they have to take into consideration how much nitrogen is going to be taken up by the crop itself. So they put some extra in. I don't think that's necessary for a specialty. I don't think that's what they do so much for specialty crops.
Of because this carbon relates to the biomass. The microbes obviously create the microbiomass. And so all of this just ties in so tightly that it makes sense for people to understand much better than I've just explained it, how to use these numbers, so to speak. So you get a 200 number or a 600 number in Anchorage, Alaska. So what if I don't have enough carbon, if I don't have enough nitrogen, if I don't have enough phosphorus? Does that number mean anything? What does that 600 number mean to me?
Yeah. So you have to, it's like us. You can give us lots and lots of sugar, but if we don't have any of the vitamins or things we need, we can't grow with it.
So
we basically malnourished.
A number of microbes. Let's say you're in Biloxi, Mississippi again, and you get a 200, and then you apply. You get some compost from Kiss Organics, and you apply it, and you come back and you take another test a couple of weeks later, and you're at 500. does that indicate to you that you've got good?
200, 500 compost.
Oh, wow.
Okay.
Yeah, you did a good job.
So
You really boosted them up. Yeah, yeah. I mean, that would be a huge amount, but we routinely see even six-fold increases when people put the right cover crop down, because the cover crop is actually being put there
okay.
to feed the microbes in the soil.
What a couple of people have asked me is, does it make sense to measure? I mean, is there a mathematical purpose? And maybe Brady, you can help.
Yeah, I
Sure.
kind of have something I'd like to throw in there. All right, so there's this scientist in Europe. What country? Heidi Anderson?
Yeah.
Germany?
They're German.
Okay. So these German guys who invented ways of measuring microbial biomass, but they're long and drawn out, but they're really quite good. And they did correlations with. I've compiled data from a couple of papers. I have 147 soils from all over Europe, farms, forests, everything. And so they wanted to see what the correlation was between microbial biomass and the actual available nitrogen in the soil.
Right.
And the available carbon. So when you send your soil out to a lab and they do a nitrate test, that's a very volatile number, because that stuff is not stable. It can wash away or evaporate. So what they were looking at was what's called the bioavailable nitrogen, and that's a totally different test. It's a test that people use to figure out the protein content in food. It was invented in 1883, and it's very expensive. So what I'm trying to I've got a lot more research to do, and Judy and I, will be discussing this, but if we can take the microbial biomass that we determine through the microbiometer and then tell you how that would relate to your bioavailable nitrogen or your bioavailable carbon, then you might have to add some chemical fertilizers, but you'd have a number based on the microbiometer reading that would inform how much you really need to add.
Yeah, but
like you talk about stoichiometry, the ratios.
I mean, what they showed was that the ratio of that nitrogen, the available nitrogen, correlates very highly, R squared over 0.8,
right.
with the microbial biomass.
That's why this test is such an important test to me, because it tells me that if I see a change in number because somebody adds a product into it, that is increasing the biomass and increasing nutrient availability.
Yeah, because, you pointed this out, I think, last time, and it's in your books too, which I'll recommend again, okay, that there are organisms that fix nitrogen from the atmosphere in the soil, not only in the nodules that can inhabit plants and are dependent there, but some of these live within the soil, and they are creating nitrogen. And you see that when you have a large and diverse population, that these are working. So the laboratory tests people get are telling you what they can measure chemically. They're not telling you what actually is there available to the plant.
It's like what they do is they rinse the soil and see what comes out in the water, right? And then, but this bioavailable nitrogen is stuff that is stuck to the soil in long chains and can be, excavated by the microbes.
It's sort of like when you eat. Not all of it's used for nutrition. Some of it you got to get rid of, and some of it's available and some of it isn't. That's just all there is to it. Or let's put it in a fungal way. If you eat a mushroom without cooking it, like so many of us have all throughout our lives, in every salad that you get at a fancy restaurant. If it's not cooked, it goes right through you. It's not available. But if you cook that mushroom, oh my goodness gracious, the vitamin D and all that other stuff you get. So it's the same thing. It's the same thing here. But back to these numbers. So, but why is there such a difference? People always want to know, between my number in Alaska, where we've got terrific soils, and the number in Mississippi, where the soils aren't so good. And you've attributed it properly to the ability of the soil to be able to hold nutrients, and to be able to nutrients to be able to be broken down and to be able to be held in the soil, as opposed to not being broken down and being washed out in the soil. When someone sees a drop,
but also your soil up there can only be used for 2 months of the year, or for a long period of time it could. So it's not been exclusively mined by crops the way
Right.
the soil
That's
in
exactly.
the rest of the United States has been.
That's a good
And
point.
you haven't lost soil either. In the Midwest, they have lost 3 feet of
Topsoil.
Yeah,
topsoil
I think it's up to an
in
inch
the
a year now,
last 100
which
years.
is really,
I mean,
well, and all anybody has to do today, of all days, the Sierra Desert is blowing, past Miami into Houston, and the same thing happens, from the fields, prairie, what used to be prairies that are now farm fields, in Illinois, when the wind's blowing in the other direction. So it is a serious problem. And again, the concept is, you've got the right carbon nitrogen ratio to support the microbial life. They create literally a microbial web that holds that soil. They create the slime, they create all that structure. So it's all key stuff. When we talked about this stuff, I guess originally, and people were testing it, did you have a concept that you'd get sort of different readings in different places in the world? I mean, was that something?
I mean, we had read a little bit, but I was never so aware of the very big differences throughout the world, and I think, we're really only learning a lot about this now, especially when people are talking about carbon sequestration. One of the big issues is, like, how much can you actually store in certain soils?
Mhm.
Like, what is a reasonable goal? And how rapidly can you do this? But I think, this week we quoted Kirkby, and he's been very, very successful. His group has, in really increasing the amount of carbon that's stored in the soil. And they do use, extra nitrogen, phosphorus, sulfur ways. They try, I believe, to supply it with, with dung, with animal fertilizer. But, people have successfully used fertilizers to up a non-nutritious green manure. And you can do, if you do that, like watching the number of microbes, if you do it in such a way that you don't decrease the microbes, you're in good shape. But if you put so much down that your microbes don't go up to 200-300, then you're in trouble. In the really big chemical ones, we never see above 100.
What about when people do individual pot culture? We got a lot of cannabis growers who use the microbiometer. I mean, can you distinguish between, okay, this is pot? In other words, I guess what I'm getting at is, when you have an ability to be able to see what kind of soil type people have across the world, you don't know whether they're using Kiss Organics compost in a pot, and growing beautiful cannabis, I might add. And I'm giving Kiss Organics a little plug, because Todd is such a great guy, and I love his father and his mother, and he's just a great guy. And I can only imagine. Yeah, but anyway.
I mean, I certainly recommend sticking with organics, but I know, like in Australia, they have some particular problems, and a lot of farmers are up. If it's a farmer, he's up against the fact that he has to transition slowly, or he's going to lose yield. And if he loses yield, he's working on a 1% a year margin. He can't afford to have his yield go way down. So while he's trying to build his soil up at the same time, he's also trying to keep his yield up. So it's a big problem.
Why indoor growers and a lot of people, even outdoors, grow stuff in big pots. So is there going to be a way to be able to tell you, this number's from soil in a pot, even though it's from Australia, or if I'm in Biloxi, Mississippi again, and all of a sudden.
Well,
okay.
It would be the user has to offer that information. Like so right now there's questions. You take your sample and then there's questions after the fact, right? Only thing I ask beforehand is it soil compost or compost tea, which I'm also going to add compost extract to now.
I agree.
Because we need to be able to differentiate. And if we have that, and then we could ask about growing environment and the base of the soil. Is it engineered? Is it compost? That sort of thing. Yeah, then that gives us even more information to give ranges to our users.
Because again,
that would help them.
GIS, graphic information systems, which are all over the place now. It's interesting because I'm sure the USDA must have a graphic information system that shows you your soil type throughout the country. I am sure there isn't but there isn't one that shows you the biomass microbiome throughout the country, because of the standard. There's a standard system until you guys came along. It was cheap enough for people to take the reading and put it into a database. This is all going to be citizen science, ultimate citizen science.
Right. Yeah. Well, what I've done now is I've taken all those points on that map and I've assigned a microbial biomass to them, and I'm working on getting that plotted so you can spin it around and see,
interesting,
what areas have some of the higher readings.
You got to continue to do research on, exactly what does, microbiomass do. I mean, we know what it means, but what does it do? So, for example, something that I've always am confronted with, and I give one answer, and maybe I should start giving the answer in a microbiomass way. People say, okay, I'm a soil food webby, but my neighbor is a Korean natural farmer. A soil food webby uses aerobic conditions to break down materials. But there's all sorts, and in my books I say, oh, you don't want to go anaerobic. Anaerobic is bad. You get E. coli. You get all the bad things if you go anaerobic. You Dr. Judith, understand that because you were in the medical field. Anaerobic is what breeds bad stuff. But Korean natural farming and EM and all that kind of stuff. What I say to people is it's using an anaerobic system to break the material down so that it can then be used aerobically in the soil by the microbes. The microbes aren't used.
Well, there are crops that are raised anaerobically, like rice. So that is raised underwater. So obviously it's an anaerobic system. So I feel bad for the anaerobes. They get such a bad rap. They're not all bad.
But you want your soil to be. First of all, I got to go back and think about rice because I always thought there was a little oxygen involved there. But anyway, there are anaerobic pockets in the soil, but in anaerobic situations, you produce alcohol, and you don't want your roots growing in alcohol soil. So when you take your bokashi, the use of the bokashi is to break down material so that it is then usable by the lower levels of the bacteria, the bacteria who ultimately make it available to the plants, et cetera, et cetera. But they don't do, it's not anaerobic bacteria that you're looking for in the soil. And so I think that's something I have to correlate in terms of biomass. Is there a difference between?
I think we're talking about like what are the conditions that mean that you're growing an anaerobic group. Okay, so the anaerobes that are growing in rice soil are the healthy anaerobes, and that's good soil, and that's what they require to grow. So, but if you take a completely different environment and it's suddenly invaded by a bunch of anaerobes which don't belong there and it's not their habitat, then you're talking a completely different story. So a lot of it is the environment that they find themselves in, and whether they're natural in that environment or basically invaders
clearly.
because
but
of changed conditions.
Aerobic as well. I mean, if they're in the wrong spot, they can. But so what I'm trying to figure out is using the microbiometer. I haven't used it on anaerobic composting situations. The reading. I mean, I would get a microbiometer reading off of a sample of bokashi, and it might be very, very high, but that might not be necessarily something I would want to put directly on my vegetables. Is that possible? In other words, when I take a microbio,
well,
if it's an anaerobic sample.
Go ahead. You've taken the microbiome.
Well, if something's anaerobic, we measure the microbes. We can't tell whether they're aerobic or anaerobic. Okay, but what we hypothesize when we dry the soil out, okay, and then we reconstitute it. If it's been anaerobic, it doesn't come back anywhere at all, practically.
Right.
So because when you dried it, you've taken a real anaerobe, and they can't survive in oxygen. Okay, so you've actually just killed them off. They die. I mean, if they want to measure an anaerobe from your body, they have to take it under anaerobic conditions, and they have to grow it under anaerobic conditions. It's cultured in a hood that has no oxygen and everything.
Well, I mean, but wouldn't that anaerobic compost go right along with your Korean friend and the way he uses anaerobes to break down things? I mean, you're breaking down this green manure to make finished compost. And by the time you're done and you've turned it, you've killed off the anaerobes, and now you've just got the aerobes left.
That's exactly right. And that's how people say, well, what's the difference? And to me, there is no difference at the end product. It's how you break the material down. And what you're saying,
well, my understanding is alcohols and the polyphenols and some of this stuff, that are intermediate from anaerobic breakdown, and that is what hurts the soil and the plant. So as the compost matures, then those compounds are converted into non-toxic compounds as the compost matures.
Compost becomes aerobic.
Right.
I mean, it's compost. So
right.
And so the point, I guess the point is that this could be a tool that we can use for anaerobic methods as well as soil food web methods. And if there are people who are listening now who are using Korean natural farming methods, etc., it sure would be interesting for you to run some experiments using bokashi EM, to see how this all correlates and helps your kind of growing, because I know it tells me things team with microbes that are aerobic, and I think what we've had is a confirmation that it'll also tell you whether it's teeming with microbes, anaerobic as well. So people should, that would be an interesting sort of branch of studies that we need to do in this citizen science that we call, you know, the microbiometer. Does that make sense? Well,
that makes a lot of sense.
Makes a lot of sense, yeah.
I think we should probably. I think we should probably
I think
say
so.
good night, good afternoon, whatever.
All right.
Good morning to you in Alaska.
You know, wrapped people's heads around a little bit. We're trying to get the, you know, the concept of microbiomass and nutrition and carbon to nitrogen ratios, and we'll have some more talks about this. You know, maybe next week or the week after. What do you think?
Yeah, I think so, and I think, you know, I recommend to people who are really interested in this that they read your book, Teeming with Nutrition. Okay, very, very important. Okay, to get a better understanding of this complex subject.
And of course, I always tell people that microbiometer, www.microbiometer.com, tells me whether products teem with microbes, tells me what stage my plants are going in. It's a phenomenal tool. Yeah.
Okay, and right now, if someone orders a starter kit for $5, they can get we'll send them with that starter kit a copy of Teeming with Microbes. Yeah, because we know that it's understanding what the data means that you're getting is what allows people to use this tool.
www.microbiometer.com. We're going to do some citizen science, folks. That's what this is all about. There's some real science going on at Cornell and other places on the microbiometer, and we're going to get back with Judy and of course with Brady, and maybe even some of the other folks at the microbiometer company. www.microbiometer.com and teeming with microbes and all. Oh, and don't forget that one. All right, enough of this stuff. Thanks, folks. See you in a few weeks. Yep. Bye now.
Okay.
Be good, Jeff.
Have a good one.
Bye, everyone.
Bye.